Good day, Ma'am Helen Vicente, esteemed members of Thesis community, distinguished guests, fellow students, and all those who have gathered here today. I am deeply honored and privileged to stand before you to present my thesis presentation of Evolutionary History of Angler. The Theloist order Lophiformes, commonly known as anglerfish, contains a diverse array of marine fishes, ranging from benthic shallow water dwellers to highly modified deep sea midwater species. They comprise 3 to 1 living species placed in 68 genera, 18 families, and 5 suborders, but approximately half of the species diversity is occupied by the sea ceratoids distributed among 11 families. Lophiformes obtain a comprehensive picture of the phylogeny and evolutionary history of the anglerfish. We assembled whole mitochondrial genome sequence from 39 Lophiformes, 33 newly determined during this study, representing all 5 suborders and 17 of the 18 families sequence of 77 Hyteloist. The anglerfish, called as Lophiformes, is a fascinating order of bony fishes with remarkable diversity ranging from benthic ambush predators in shallow waters to highly modified. Their bizarre adaptation, including lures, bioluminescence, and sexual dimorphism, raise captivating questions about their evolutionary history. However, understanding their past has been challenging. These are the example of anglerfish, representatives of the Lophiform suborders Lophioids, Antennarius commersons, Semterectis, Chonax caruso, representative of the Lophiform suborders. Reconstructing the evolutionary history of anglerfish, a diverse and morphology complex group of bony fishes, has been challenging to an incomplete fossil record and ambiguous relationship based on morphology alone, which means other studies indicate that anglerfish only originated shortly after the extinction event as part of the massive adaptive radiation of Percomops. Although this clashes with the extensive diversity already known from the studying the evolutionary history of anglerfish from a metagenomic perspective holds several significant implications and distribution of the broader fields of evolutionary biology, ecology, and conservation. Here are some key points of highlighting the significance of the study. Phylogenetic understanding. This means understanding the evolutionary branching patterns helps elucidate the evolutionary history of this group, clarifying the relationship with other fish species and contributing to the broader understanding of Theloist fish evolution. The biogeography and historical biogeography. By tracing the evolutionary history of anglerfishness, researchers can gain insights into the biography of this group. Divergence time estimation. This data can be used to estimate the timing of key evolution events, providing a temporal framework for the evolutionary history of anglerfish. The general objectives of this study is to aim to provide a comprehensive and well-supported picture of anglerfish evolution, revealing the relationships, adaptation patterns, and diversification history. This research also offered valuable insights into the unique adaptation of this fascinating fish and contributed to our understanding of evolution and adaptation in deep sea environments. Here are some examples of specific objectives that researchers might consider. Sequence metagenomes. Obtain complete or near complete metagenic sequence for representative species of anglerfish within the Lophiformes order, which means you need to focus on the goal of representative of all the species of anglerfish to near complete metagenic the sequence. And also the phylogenetic analysis. Conduct phylogenetic analysis using the metagenic data to resolve relationship among different genera and species of anglerfish. The divergent time estimation is an integrated fossil calibration points or other external information to anchor the molecular clock analysis. And the biogeographic reconstruction is to explore the historical biography of the anglerfish by incorporating geographic information into the phylogenetic framework. And we also have adaptive evolution analysis is to identify and analyze specific mitochondrial genes associated with adaptation unique to anglerfish such as those involved in bioluminescence or deep sea adaptation. And the last is interdisciplinary integration. Integrate metagenomic data with other available data sources such as morphological traits, ecological characteristics, and geological information. And this is my conceptual framework. Intrasubordinal relationship and intersubordinal relationship. The review or related literature. Anglerfish, or we call genus Liphus, have an affinity for the relatively warm water masses of mid-latitude. The anglerfish species Lophius piscatorius in the northeast Atlantic is a boreal fish, which generally is distributed between the Straits of Gibraltar and Western Mediterranean Sea, routed by Caruso in 1983 in the south. Culturing and maintenance of anglerfish in artificial conditions are not common practices. Researchers and aquarists typically focus on the deep sea organisms that are movable to captive condition and have potential application in research, education, or public display, which means anglerfish inhabit the ocean at deep of 0 to 1,000 meters, rarely dropping below the continental slope. This wide range of depth is influenced by fish age and seasonality, water temperature, and prey availability. They lie half buried in the sediment, sand, or mud as they wait for their prey as continental shells. The anglerfish mating behavior is fascinating and truly unique example of sexual dimorphism in the animal kingdom. Here's the breakdown of their underwater courtship dance. The female anglerfish is a behemoth, dwarfing the male by several times. Her bioluminescent lure acts as a beacon or what you mean call or bait to draw in the tiny bioluminescent males who mistake it for a meal. If the male approaches, drawn by lure's irresistible glow, but instead of becoming a snack, he latches onto the female's body, teeth digging into her skin, and the male body degenerates, fusing with the female, providing her with nutrients and sperm. Essentially, he becomes a parasitic mate, sacrificing his independence for productive success. The female anglerfish can carry multiple males, each dangling like tiny lanterns on her body, providing her with sperm for future fertilization once their usefulness is over. The male bodies are absorbed by the female. Morphological comparison of anglerfish. Morphological comparison in the context of the evolutionary history of anglerfish from metagenomic perspective involve assessing physical characteristic and the structural feature across different species. We explore this idea through an analysis of body shape evolution across ocean depth zone in over 2,000 species of marine theolist fishes. We find that deep ocean contains twice the body shape disparity of shallow waters driven by elevated rates of evolution in traits associated with locomotion. As we can see in the picture, shallow, intermediate, and deep. These are the fish that can survive in this depth. In summary, the metagenomic perspective on the evolutionary history of anglerfish provides a comprehensive understanding of their genetic heritage, adaptation, and relationship. In 2010, mitochondrial genome phylogenetic study suggested that anglerfish diversified in a short period of the early to mid Cretaceous. Between 130 and 100 million years ago, a more recent preprint reduced this time to the late Cretaceous between 92 to 61 million years ago. This is my recommendation if you are conducting research. If you are considering pursuing research in anglerfish, especially in the field of marine biology or deep sea ecology, here are some recommendations for future researchers. Build a strong educational foundation, which means it's a solid education in biology or related field. A strong foundation in biological science will be essential for understanding the complexities of anglerfish biology. Specialize in ichthyology and deep sea biology. Consider specializing the study of fish and deep sea biology. Courses and research opportunities focused on these areas will provide you with necessary knowledge and skills. Stay informed on recent research. You can compare your research to your review related literature, keep abreast of the latest research in anglerfish biology, subscribe to scientific journals or attend conference and follow the work of leading researchers in the field to stay informed about recent discoveries and advancement. Implication: future researchers in anglerfish have the opportunity to contribute significantly to our understanding of this fascinating deep sea organism. Their work can have implications for various scientific, ecological, and conservation related aspects. Here is a potential implication for the future researcher in anglerfish. Future researcher can delve deeper into the evolutionary history of anglerfish using advanced genomic techniques. Understanding the evolutionary relationship, divergence, and adaptation of their species can provide valuable insight into the broader context of deep sea evolution. Another recommendation: develop proficiency in molecular techniques. Given the importance of metagenomics in the study of anglerfish, develop proficiency in molecular biology techniques. Familiarize yourself with DNA sequencing, PCR, or what we call polymerase chain reaction, and bioinformatics tools used in genomics research. Collaborate with experts. Collaborate with researchers and experts in the field. Building a network with experienced scientists will provide valuable mentorship, guidance, and potential collaborative opportunities for your research. To make discoveries, academic research is usually thought to mean an equal partnership between two academic faculty members who are pursuing mutually interesting and beneficial research. Utilize technological advance. So, as we all know that this generation these days is so high tech. Stay updated on technological advance in marine research such as remotely operated vehicles and autonomous underwater vehicles. These tools enable researchers to explore deep sea environments and study organisms like anglerfish in their natural habitats. Researchers would like to express their sincere gratitude to everyone who helped with the accomplishment of this study project successfully. Firstly, I would want to express my gratitude to the omnipotent and all-pervading Heavenly Father who has been their source of wisdom and understanding during this scholarly pursuit. To my closest friends and families who have always been my pillar of support. My heartfelt thanks to Professor Helen Vicente for their assistance in fieldwork and laboratory analysis. Their dedication and expertise were instrumental in the collection and processing of data crucial to success of this study. The original authors of this research study are to be thanked and acknowledged for the content of their research. Their unparalleled efforts to ensure that this report was presented in its entirety and spoke about their research.